So fungus can reproduce either sexually or asexually. Molds are the type of visual fungus that you see if you leave your fruit or bread out on your counter for too long. So typically, spores can be made sexually or asexually if a spore is carried a long distance. So spores are tiny, microscopic single cells that will lead to a fungus, and they travel through the air and on the wind very easily. And if you leave your fruit out on the counter for a very long time, they will definitely get fungal spores on them and then eventually start to mold. And those molds that you see are the visible mycelium. So you can see the mold, and then here's the close-up mycelium with all of these hyphae. And then they're typically colored. So you can see these little spores that are being made. And this is asexual, just made through mitosis and cell division. And then they can release these spores into the air in your kitchen to get more spores on the rest of the food you leave out on the counter. Keep in mind that molds can reproduce sexually as well, but they also can also reproduce asexually. Yeasts are those single-celled fungus, and they usually reproduce asexually. So this is going to be through cell division, just like mitosis. They're going to make clones. And so usually what happens is the parent cell will actually pinch off a budding cell. So you can see a little bud starting right there. Here's a larger bud. And then this is, you can see these two cells are pinching off from each other to make two new cells. So early mycologists, so mycologist, myco means fungus, ology is study of, and a mycologist is someone who studies fungus. Early mycologists typically classified fungus based on how they reproduced. So what you'll see in the classification section is that certain clades or groupings of fungus typically all reproduce in a similar way. And that's how we distinguish between them. Now we don't use that extensively. We also use the genetic sequences, and so some of our groupings turned out to be monophyletic clades because they were backed up by genetic evidence, but some are also polyphyletic or paraphyletic because the genetic evidence doesn't really support it as a clade. But we typically still group fungus by how they reproduce. There are about 20,000 species of fungus that are known to only reproduce asexually. And that doesn't mean they don't have a sexual stage, it just means we don't know about it. So those are typically called deuteromycetes. Deutero means second. Mycetes is the term you're going to see for all our fungal clades. And typically these fungus are called deuteromycetes. This is definitely not a clade, and they're just grouped together because they reproduce asexually. And if a fungus that's considered a deuteromycetes, we eventually learn that it has a sexual stage, typically it'll be reclassified to whichever group it fits in best. So fungus reproduce sexually, and we're going to find that in the different groupings of fungus, they have different structures that they make to reproduce, where they make spores and what kind of spores they release. There are two types of mating types. So fungus are not going to be male or female like animals. Instead, they have different mating types. Often we just call these like plus or minus. And they use pheromones, which are hormones that they can release out into the soil to communicate to the opposite mating type. So a plus mycelium might release pheromones to attract a negative mycelium to grow towards it. And then once they grow to physically contact each other, they're going to have fusion. And there are two words here that you absolutely have to know for the exam: plasmogamy and karyogamy. So down here we've got kind of a generic fungal asexual life cycle and sexual life cycle. So let's go through this sexual life cycle, and then what we'll do when we go through each group is you'll see that there are slight differences on this one plan. So to start off mating sexually, you need two individuals. So we might have a plus type and a minus type, and they're going to grow toward each other thanks to their pheromones. And then eventually they're going to come into physical contact. And when they come into physical contact, the cytoplasm is going to fuse together. So this is called plasmogamy, is when the cytoplasm fuses together. All fungus, their mycelium, are haploid. So typically, we've already got haploid cells. So when they fuse together, we now are going to have cells that have, so here's my hyphae, my cells, they have 2 haploid nucleuses. So this is N and this is N. So the entire cell is 2N, it's diploid, but it's not fused yet. They're just both existing in the same cell. So here would be the next cell, 1N, 2N. So where maybe this one came from the plus and this one came from the minus. So this is plasmogamy, where the cytoplasm of 2 different individual mycelium has fused, but we still have separate nuclei. And this stage is called heterokaryon. So hetero is different, and karyon means nucleus. So it literally has 2 different nuclei inside of there. And when it looks like this, it's called dikaryotic when they pair off into individual cells. Eventually, these 2 nucleus will fuse in a process called karyogamy. So karyo again means nucleus, and then gamy is the fusion. The amount of time that it spends in this heterokaryotic stage can be hours. So this tends to be anywhere from hours to days to even centuries. So a mycelium can exist in this heterokaryotic stage for a very long time. Eventually, if it reaches maybe a environment that's not very friendly, where it needs to create some sexual diversity and some new spores, it's going to go ahead and do karyogamy, where these 2 nuclei are going to fuse. And for a very short period of time, we're going to have a single nucleus that's 2N inside of each of these cells. This process here is very fast. So once this fusion happens, they're going to go right into meiosis and start making haploid N spores. And then those spores are going to typically germinate onto some sort of fruiting body like a mushroom, and then be released into the air to go make a new mycelium somewhere else.